Okay, can you see? One of you can unmute and tell me, can you see this
Yes,
diagram?
ma'am.
Yes, ma'am.
Yes,
What
ma'am.
is that? Can you read the heading of the
Yes,
figure?
ma'am.
What is that heading?
Misuse of
Urine formation. Okay. Okay. Now I just came to this diagram purposely because I until and unless you understand which part functions how, so that you will understand the remaining part. Okay. Now listen. See, this part which is called as Malpighian body, which contains the Bowman's capsule and the glomerulus. So here, whatever the filtration is done, because of the difference between the diameter of afferent and efferent arteriole, that is called glomerular filtration. Okay, so whatever the filtrate comes, it comes in the cavity that is called as capsular cavity or the capsule cavity. So here it comes that filtrate through this glomerulus, and that is what called as glomerular filtration. Okay, this filtration where it takes place, that is glomerular filtration. Now it enters through the neck in the PCT, that is proximal convoluted tubule, and then, as the proximal convoluted tubule is highly coiled structure, the flow of filtrate becomes very slow. And then from there, it enters, that filter enters to, or it reaches to the loop of Finlay, which shows the two limbs. One is the descending limb and one is the ascending limb. Okay. So this is, which is downwardly directed, that is descending limb, which is upwardly directed, that is ascending limb. It is shown also. Now one more thing you can observe, that the part of thickness of the descending limb and ascending limb, as well as the proximal part of the ascending limb, which is thinner, and the distal part of the ascending limb, it is thicker. Then one more thing you have to remember, that is the part of descending limb which is permeable to the water, but the part of the ascending limb of the loop of Henle, that is impermeable to water but permeable to the electrolytes or the ions, you can say, and this helps in making the urine concentrated. Now what happens, again I will repeat, when the filtrate reaches the capsule, from the capsule through the neck region, it reaches the coiled structure of PCT, where the flow of filtrate becomes very slow, so that maximum reabsorption from the filtrate takes place. And as this part, that is the when it reaches here, the maximum reabsorption of the water takes place here in the PCT region, as well as in the descending limb of the loop of Henle also, maximum reabsorption of water takes place, because that wall of descending limb is permeable to water, whereas the wall of ascending limb is impermeable to water. So here the reabsorption of ions takes place. Only electrolytes it is permeable, not to the water. So what happens when the filtrate comes through this proximal convoluted tubule to the descending loop of inlay, maximum reabsorption of water takes place here through this wall, as it is permeable to the water. So that what happens, this urine, that means the filtrate, which is called as primary urine, it becomes concentrated, because maximum water is reabsorbed by the walls of PCT as well as descending loop of Henle. And as it reaches through the, taking the U-turn to the ascending limb, then this thicker part, and this ascending limb being impermeable to water, only it is permeable to the electrolytes, so that only the ions like NaCl only can be reabsorbed or they can be passed out. Okay, so this then reaches, so this urine becomes concentrated, and then it reaches to the DCT, that means distal convoluted tubule. Now this distal convoluted tubule is also coiled part, but the walls of the or the cells of the walls of this DCT are short, and they do not show any increased area like microvilli in this region. So reabsorption of water doesn't take place in this region, or the substances, the reabsorption of these substances doesn't take place here in this region as compared to PCT. PCT, the walls are inner walls layers of the endothelium wall or the layer. It shows the presence of microvilli, which shows gives the appearance of brush border, which is helpful in increasing the surface area, which helps for the maximum reabsorption. So maximum reabsorption takes place here in this region in the loop of Henle, and then when the urine, which is now concentrated, reaches DCT, here water cannot be reabsorbed, but the ions like urea, some of the substances like urea can be released in the released out, as well as and when required they can be reabsorbed, depending on the concentration of urea, as well as this wall or this area which is highly covered by the blood vessels. So that's why I will show you. This is what the blood vessels, or this portion is called as vasa recta, which covers this loop of Henle. That also helps in this functioning or the process of urine formation. And so in this region, as the cells are not showing any specialized structures like microvilli, reabsorption of water doesn't take place in this region, and then through this DCT it is reached, that urine reaches to the collecting duct. So this is the common area where is called as the that urine is taken, or it reaches the common duct, that is the collecting tubule. So here, as and when required, then only reabsorption of water or the ions takes place. But in this area, urine, from the urine, urea can be reabsorbed or passed out depending on its concentration. And this takes place in the with the help of these blood vessels surrounding this area. So by the simple diffusion is what? The movement of the particles or the ions or the molecules is from higher concentration to the lower concentration. When urea concentration is high in this region, then it is diffused out of this renal tubule, absorbed by the blood vessels or the fluid tissue. And when the urine concentration in the blood tissue or bloods, it becomes more, that is again diffused. Then again the urea concentration is increased in the tubular region, that means the renal tubule region. So here this is called as urea cycle, because by the simple diffusion or process, the urea concentration or the urea is diffused from the renal tubule or the tubular region, or can be absorbed again. So this takes place in this region, that is the DCT region. And at the end, that urine is collected, or it is taken, or it reaches to the collecting tubule and then to the common duct. So this is what, again I repeated, the process of urine formation, which is going to help you for the next part. But before that, this equation you try to understand. What is this equation? EPF, which is equal to hydrostatic pressure in the glomerulus minus osmotic pressure of the blood and filtrate hydrostatic pressure. That means what? See, this is what glomerular filtration I told you. Whatever the filtration takes place, all the small particles, small size particles are getting filtrated here, and that plasma without proteins, only proteins and few particles like blood cells, they are large particles, large size molecules, so they cannot be filtered. So this filtrate is considered to be glomerular filtrate. Okay, and what is then tubular reabsorption? Now, whatever is reabsorbed from the tubular fluid back into the blood, that is called as tubular reabsorption. Try to understand this now. From the neck region up from PCT up to the common duct, the entire part is called as renal tubule, and whatever the filtrate comes in this renal tubule, that is reabsorbed again to make the urine more concentrated, and that is called as tubular reabsorption. So the movement of the substances from the tubular fluid back into the blood. Why back into the blood it is told? Because the entire area here, it is covered by the blood vessels, isn't it? So that's why this is called as this, the walls, the cells in the walls of the PCT or in the tubular region, they are having this capacity to reabsorb. So maximum substances are reabsorbed. Here water is reabsorbed in this region, here ions are reabsorbed and released. So the movement of the substances from the tubular fluid back to the blood, that is the in the blood, that is called as tubular reabsorption. So tubular reabsorption takes place in the PCT and loop of Henle. Okay, now the next is what? Tubular secretion. So the movement of substances from the blood into the tubular fluid is called as tubular secretion. Just now I told you. So this tubular secretion takes place in the DCT region and to some extent in the CT region, that means distal convoluted tubule, and to some extent collecting tubule, this region. How? Because here the movement of substances from blood, that means which surround these this renal tubule are surrounded by blood vessels. So from the blood again it is released, secreted as and when required in the blood, in the tubular fluid. That means into the fluid which is, that is considered to be the urine, that we call it as urine primary or whatever that filtrate. So again these are secreted in the filtrate. Just now I told you before this that the urea which is reabsorbed again from the blood vessels, and again if concentration of the urea in the tubular region increases by the simple diffusion, that is from higher concentration to the lower concentration, that gets diffused in the blood vessels, and if it again the urea concentration in the blood increases, again it gets diffused in the tubular region, that is the tubular fluid. So this is what the reabsorption and the secretion. So this is what called as secretion. What is secreted from the blood? Urea is secreted. I'm giving just one example, but some ions like Na, K plus or Cl, such ions are also reabsorbed as and when required to make the urine more concentrated, and so that this is what tubular secretion. So what is this tubular secretion? From the blood vessels, the substances like urea from the blood are diffused or moved or transported again or reabsorbed again or secreted by the blood cells into the tubular, that is the renal tubular region or the renal tubule region, that is what called as tubular secretion. So tubular secretion is taking place in this region, that is called as DCT and CT region. So all the three steps, they are taking place at a particular part of the nephron. So glomerular filtration takes place in the glomerulus part, that means the Malpighian corpuscle, this area, that is called as glomerular filtration. Tubular reabsorption takes place in TCT and loop of Henle, and then tubular secretion takes place in DCT and CT, that means distal convoluted tubule and collecting tubule. And then that whatever the name we call at urine, it is collected in the this collecting tubule, and which is present at the end, it is collected, it comes in the pelvis region of the nephron, that is the internal part if you remember, and then from here there it is collected by ureter and passed to the urinary bladder, and through urethra it is thrown out of the body. So whatever urine is produced, it is more concentrated because though it contains it, so here though the filtrate contains so many substances, 85 to 90%, then it is filtered here. So it then at the end the urine which is produced, the concentration is very high as compared to this glomerular filtrate concentration. So that's why the EFP, that means the entire efficient, or we can say filtrate pressure, is equal to hydrostatic pressure in the glomerulus. That means this, whatever the pressure is created because of the change in the size of the afferent and efferent arteriole, this is what called as glomerular filtration. That is this minus, what is that? Pressure. This is the pressure of the glomerulus, that is hydrostatic pressure, because of the difference in the afferent and efferent arteriole. Now minus osmotic pressure of the blood, means whatever the blood it's inside the capillaries, it is having its own pressure, that is called as pressure of blood, that is osmotic pressure of the blood, plus filtrate hydrostatic pressure. That means once this is filtered, this cavity, whichever it shows the capsular space, which you can see, that filtrate, it is also having its own pressure, that is called as hydrostatic pressure. So
Excuse
EF,
me, ma'am.
yes.
Ma'am, your screen is not visible.
Okay, wait, I will come back again. Okay, you can't see anything. From which part, you just tell me, so that I will unmute yourself and answer. I'll stop sharing and again I will go to presentation mode. Can you see now?
Yes,
Yes,
ma'am.
ma'am.
Up to what you were able to see the screen? I have explained the entire process of urine formation again, and I showed you the parts also, where which part is involved in glomerular filtration, which part is included in the tubular reabsorption, and which part is included in the tubular secretion. So, uh, have you seen that part? Were you able to see and listen? Unmute yourself first. Tell me, is my voice audible right now or no? Shreya or Shraddha, whoever is there.
Yes, ma'am. Your voice is clear.
Okay, and is this clear now screen? Is it visible?
Yes, ma'am.
Ah, now I will repeat this part again, that glomerular filtration takes place in this region, that is the Malpighian corpuscle, and tubular reabsorption takes place in PCT and loop of Henle, whereas tubular secretion takes place in DCT and collecting tubule. So what does it mean by hydrostatic pressure in glomerulus? The pressure which is created because of the difference in the afferent and efferent arteriole, that is called as hydrostatic pressure in glomerulus. Okay, this minus osmotic pressure of blood and filtrate hydrostatic pressure. Now what is the osmotic pressure of blood? Now whatever the blood capillaries are carrying blood, it is having its own pressure, blood is having its own osmotic pressure, so that is called as osmotic pressure of blood. And whatever the filtrate comes here, that is the in the capsular region, that filtrate also is having its own hydrostatic pressure, which is um, uh, uh, forcing, creating a pressure against this. So this is what called as filtrate hydrostatic pressure. So EFP, that is efficient filtrate pressure, or the entire filtrate pressure, is what? Hydrostatic pressure in the glomerulus minus osmotic pressure of blood and the hydrostatic pressure of filtrate. So this you learn. So this is equivalent to 10. How? Because this is 55, or in some textbook it is mentioned as 60 at around. Then osmotic pressure of blood, it is 30. And uh, this hydrostatic filtrate pressure is 15. So 55 minus 45 is 10. So almost this entire, this pressure is considered to be, EFP is considered to be 10 mm per Hg. So this is all about the process of urine formation taking part in different parts of the nephron. Now, I have explained the entire part with the help of the same diagram, but it is shown here in the textbook with the help of another diagram. Here is the descending loop. This is the ascending loop, where up to this area reabsorption takes place, and this is the area where the secretion takes place. And this is the area where the glomerular filtration takes place. Okay, now we'll come to the next part, that is the concentration of urine. Now, I have already told you, but I will read some part and then I will uh, explain the remaining part. Okay. Under the condition like water intake or high water loss due to sweating, human can produce concentrated urine, and it can be almost 4 times concentrated, that is 1200 moles or osmoles per liter, than blood, which is 300 osmoles per liter. Osmolarity we studied that day, that is nothing but the solute concentration. Okay, for this purpose, a mechanism called counter current mechanism is operated in human kidney. That day we discussed also why it is called counter current. Because of counter is what? Opposite. Current is flow. As the blood is flowing in the opposite direction in the loop of Henle, descending and ascending limb, the urine, that means the primary urine or the filtrate, is flowing in the opposite direction. So that is what called as counter current mechanism. As well as the blood vessels which are surrounding this loop of Henle also, they are running in the opposite direction. The blood vessels running in the descending loop in case of vasa recta, as well as they are both the blood vessels descending and ascending order, they are running. So their flow is also in the opposite direction, which is shown in the here in the vasa recta region and in the nephron region. In this region you can see this is the descending limb of nephron, and this is the ascending limb of nephron, that is loop of Henle, where both are in opposite direction. They are flowing, that means urine is flowing, that is the filtrate is flowing. Whereas this vasa recta, which shows the blood vessels which surround the loop of Henle, right? So there is also in the blood flow is in the opposite direction. Here is the descending loop of vasa recta, and here is the ascending limb of vasa recta, which is again totally opposite to that of the tubular, this renal tubular directions, that is a loop of Henle. So this is what called as counter current mechanism, which helps in maintaining the pH as well as ionic percentage or the concentration of the urine. So one more thing is very important in this case, that is ADH. That means what? Anti-diuretic hormone. ADH is nothing but anti-diuretic hormone, which makes the cells in the wall of collecting duct permeable to water. And due to this, water moves from tubular fluid into tissue fluid, making the urine concentrated. As I told you while explaining this part, that this descending part is not so much coiled as compared to PCT. And one more thing, that the cells are also of short size. They do not show the microvilli, but as and when required, I told you. So like that, to make the urine more concentrated, ADH, that means anti-diuretic hormones, that is secreted by the pituitary gland, and which makes this area of DCT and CT permeable to water for maximum reabsorption of water. And that's why what happens, then urine becomes more concentrated, because maximum water is reabsorbed by this area of DCT and CT because of the action of ADH. ADH is antidiuretic hormone, which is secreted by the pituitary gland. So this is what you need to remember. Cells in the wall of deep medullary part of collecting ducts are permeable to urine. So this area which are present in the medullary region here, this is permeable to the water, permeable to urea also. So as concentrated urine flows through it, urea diffuses from urine into tissue fluid and from tissue fluid into the tubular fluid, flowing through thin ascending limb of loop of Henle. And this urea cannot pass out from tubular fluid while flowing through thick segment of ascending limb. So I told you the same thing while explaining it with the help of the previous diagram. So I will not repeat the same thing. So ADH is playing very important role in reabsorption of water in the DCT and CT region, and that ADH is useful in the recycling of urea. Now, this is very important to make the urine concentrated. So, this is the reason why in this region, urea as well as some of the ions can be reabsorbed by because of the ADH activity or ADH secretion, which is secreted by hypothalamus, or you can say by the ADH is secreted, and because of the action of ADH, that area, that is the part of the DCT and CT, becomes permeable to water and maximum water is reabsorbed in this region to make the urine more concentrated. Only this much you remember from this part, because if you go on reading, you will not understand exactly, but you just remember what I told you just now. Now, what is the composition of urine? In short, I will just want to tell you what is that RAAS, that means RAAS, and what is the role of ADH again. See, normal urine is pale yellow colored, whereas this color comes due to the presence of pigment called as urochrome. And that composition of urine depends on the food which we take and fluid consumed by the individual. So, how this happens? There are two ways in the regulation of water reabsorption through ADH, and also other by the electrolyte reabsorption through RASS. I will tell you what is RASS. You can note it down. It is mentioned in the text also. Okay. Hypothalamus in midbrain has spatial receptors called osmoreceptors. They can detect change in osmolarity, that is measure of total number of dissolved particles per liter of solute of the blood. Just remember that the solute concentration in simple. And if osmolarity of the blood increases due to any reason, such as after eating namkeen or due to sweating, in other words, due to water loss from the body, osmoreceptors trigger release of antidiuretic hormone from nephrohypophysis, that is the posterior part of the pituitary gland. Okay, so antidiuretic hormone, that is ADH, that is released by these osmoreceptors, and osmoreceptors, they release this ADH, and this ADH then stimulates reabsorption of water from the last part of DCT as well as some part of the or the entire part of the collecting duct. And because of that, what happens? It increases the permeability of the cell, so maximum water is reabsorbed by the cells of the tubular region, CT region. So this lead to reduction of urine volume and decrease in osmolarity of blood. And once the osmolarity of the blood comes to normal, activity of osmoreceptor cells decreases, leading to decrease in ADH secretion, and this is called negative feedback. That means what? Once if there is dehydration, or just now we told that these osmoreceptors are present, which detect or which give signals to the hypothalamus part, or you can say that release the ADH from the posterior part of the pituitary gland, that is called as neurohypophysis. And that ADH, when secreted or it is acting, that what happens when ADH is started functioning, then it stimulates the reabsorption of water from the DCT and CT region. So the cell becomes permeable to water, maximum water is reabsorbed, and then in this way the osmolarity of the blood volume is maintained. osmolarity of the blood when it comes to the normal, again this function is stopped. So osmoreceptors again they decrease their osmoreceptors of the cells, they decrease their activity, and then ADH secretion is stopped, and this is called as negative feedback. So this is what about maintaining the iron concentration, as well as you can say hemostasis. That is, homeostasis means what? Maintaining the internal environment. So in this way, by reabsorption of water, this work is done by using ADH, that is antidiuretic hormone. Now, in case of hemorrhage or severe dehydration too, osmoreceptors stimulate ADH secretion, and which is very important in regular water balance through kidneys. So in this way, ADH helps in maintaining the water balance of the kidneys. But in absence of ADH, what happens? Diuresis, that means dilution of urine takes place. That means urine becomes more diluted. Okay, so in that case, and person tends to extend large amount of dilute urine, which is, that condition is called as diabetes insipidus. When the person passes out the urine in large amount, and that is the dilute urine, that is called as diabetes insipidus. Another regulatory mechanism is RAAS, that is called as RAAS. Means one way of maintaining the ionic concentration or the water balance of the urine is by ADH secretion. The other way is what? RAAS regulation or mechanism. Renin angiotensin aldosterone system by juxtaglomerular apparatus. Now, this is, I know, little difficult to understand, but I will try with this same textbook, and again I will explain the same part with the help of my slides again. But try to understand what is that. When blood supply due to change in blood pressure or blood volume to afferent arteriole decreases, that means BP becomes low, or in the dehydration condition, JGA, that means juxtaglomerular apparatus cells release renin. Now you know where these are. We have already studied, discussed in the previous lectures, which are present near the that DCT cells when comes in contact with the Malpighian corpuscle region, or the Bowman's capsule region. They come in contact with that, and that region is called as JGA. Okay, so that these cells release renin. Renin converts angiotensinogen secreted by hepatocytes in liver. Hepatocytes are the cells in the liver, to angiotensin 1. An angiotensin converting enzyme further modifies angiotensin 1 to angiotensin 2, and that becomes active to form, that is the active form of hormone, which helps in the following way to make the urine its concentration or maintaining the balancing the concentration of the urine. How it acts. Now, though you will not understand this part right now, I will show again with the help of slides how it takes place. Just simply you remember that JGS is release renin, which converts angiotensinogen into angiotensin. That angiotensin is secreted by hepatocytes in liver. Okay, hepatocytes are the cells in the liver, and then angiotensin 1 gets converted into angiotensin 2 by this enzyme, that is called as angiotensin converting enzyme, that is ACE, that is also known as ACE. And this is angiotensin 2 is the active form of hormone. Now how it works, angiotensin works, what it does, what is the function of angiotensin 2? It constricts arterioles in kidney, thereby reducing blood flow and increasing blood pressure. Now try to understand, imagine now this structure, just this diagram you listen. This is the Malpighian body, corpuscle, that is the Bowman's capsule, afferent and afferent. Now what happens, because of its activity of renin, the angiotensin 2, what it does, these cells, that is the blood vessels or the glomerulus or the blood capillaries, they become narrow. Okay, so that what happens then? They constrict the capillaries. So the first function is what? Constriction of arterioles in the kidney, thereby reducing blood flow and increasing blood pressure. As the blood flow is released or reduced, automatically blood pressure is increased. And so then next point is what? It stimulates PCT cells to enhance reabsorption of Na and Cl and water. So the main function of PCT is what? Reabsorption of water. But along with that, by this activity of angiotensin 2, that activity of PCT is increased for reabsorption or the reabsorption of water as well as NaCl ions to maintain the balance of ions as well as water. Then the third point, that is the third function of angiotensin 2 is what? It stimulates adrenal cortex to release another hormone called aldosterone that stimulates DCT and CT to reabsorb more Na and water, thereby increasing blood volume and pressure. So it acts in three parts. First part is what? Malpighian body, that is the corpuscle, that means Bowman's capsule, that is the glomerular part of the Bowman's capsule. So these blood vessels become constricted, and because of that the slow blood flow becomes slow, reduced, and increases blood pressure. So this is the first thing. Second thing is the next part, PCT, where it reabsorption of water and these ions is increased. Then the third part of the renal tubule, that is DCT and CT, there also it releases the hormone called as aldosterone. That stimulates adrenal cortex to release aldosterone hormone. And what is the activity of aldosterone? It stimulates the reabsorption of these ions like Na and water, thereby increasing the blood volume and pressure. So in these three steps, in all the three regions, this angiotensin is working. First, on the blood capillaries by its construction. Second, in the PCT, maximum reabsorption of ions and water. And the third, in the by secreting aldosterone in DCT and CT region for maximum reabsorption of water and ions. So alternative functioning to ADH is angiotensin 2, that is called as RAAS. R is for renin, A is angiotensin, and the third is next A is for aldosterone, and system S is for system C. Renin, angiotensin, aldosterone system. Renin, angiotensin, aldosterone system. This is what called as RAAS. So by two ways, this maintenance of pH and ANS by the kidneys takes place. One by ADH activity, that is done in the DCT and CT region by maximum reabsorption of water and ions, and the another pathway of maintaining pH as well as ion concentration is by RAAS mechanism, that is renin angiotensin aldosterone system, by functioning, that is by constructing the blood vessels in the in the Bowman's capsule, then by maximum reabsorption of ions and water in the PCT region, and then maximum reabsorption of water and ions in the CT and DCT region by aldosterone activity. So in this way, the urine concentration is maintained. So these are the two ways, and in detail it is mentioned here, but I don't think that everyone will be able to understand, but still I will explain this part, how this ADH works here. Large, see here what happens. Action of ADH and RAS leads to increase in blood volume and osmolarity. A large increase in blood volume and pressure stimulates atrial wall to produce atrial natriuretic peptide ANP, which inhibits Na and Cl reabsorption from collecting ducts, inhibits release of renin, reduces aldosterone and ADH release too, and this leads to condition called natriuresis, that is increased excretion of Na and urine in, and diuresis. Diuresis is the dilute urine. So this is what the action of ADH and RAAS. So both ADH and RAAS are essential. Only ADH can lower blood Na concentration by way of water reabsorption in DCT and CT, whereas RAAS stimulates secretion of Na or stimulates Na reabsorption and then maintains osmolarity of fluid. So both the things are very important, and how they work, that is because of the to maintain the blood volume pressure as well as osmolarity. So this leads to the increase in the blood volume and osmolarity, and a large increase in the blood volume and pressure stimulates atrial valve, that is the heart, atrial valve, to produce atrial natriuretic peptide, that is called as ANP. And this ANP inhibits NaCl reabsorption from collecting ducts, inhibits release of renin, reduces aldosterone and ADH release too, and this leads to a condition. If this is not functioning, RAS is not functioning, or it is functioning in the opposite way, then its activity, it leads to natriuresis, that is increased concentration of Na in urine, that is ions in the urine, as well as diuresis, that means diluted urine. So this is what about the composition of urine, concentration of urine, the role of ADH and RAAS in the maintenance of ions and water in the urine composition, urine concentration. Now the last part, that is the role of other organs in secretion, only that part is remaining, and that I will switch over to the slides. So with the help of slides, I will just make it. So as I told you, urine formed is a light yellow colored watery fluid, slightly acidic, and has characteristic odor. Urine of a healthy individual contains 96% of water, 2% percent of urea, 2% of other dissolved substances, and an adult human excretes on an average 1 to 1.5 liters of urine per day, and on an average 20 to 30 grams of urea is excreted per day. Various conditions can affect the quantity and composition of urine. Analysis of urine helps in the clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose and ketone bodies in urine are indicative of diabetes mellitus. Now, what are the other organs performing excretion? First, as you know, that is the role of other organs in excretion, as you know, that along with kidney, in addition to kidney, lungs, liver, and skin help in elimination of excretory products. How the lungs perform this function of excretion, everybody, that in the form of CO2 from lungs, when you exhale the air and inhale the air and exhale the air, that during the process of respiration, what is exhaled? CO2 from lungs. So in this way, by exhalation of CO2, as well as sometimes in the form of droplets, that means water vapors from the lungs, they are excreted. So it's also significant amount of water per day in the form of water vapor in normal resting condition per day. Then the quantity of water loss increases in dry climates. Various volatile materials are also eliminated through lungs. So CO2 and water vapors are excreted from lungs. The liver. Liver is the largest gland in the human body, and it changes the decomposed hemoglobin of the burnout RBCs into bile pigments, namely bilirubin and biliverdin. So how this helps in excretion? In the form of bile pigments called bilirubin and biliverdin. So what happens? Decomposed hemoglobin of burnout RBCs that are changed into bile pigments, and these are called as bilirubin and biliverdin. So bile pigments pass into the alimentary canal along with the bile for elimination. So the liver also excretes cholesterol, degraded products of steroid hormones, certain vitamins and drugs via bile. And the last, that is skin. Human skin possesses two types of glands for the elimination of certain substances through their secretion. First is sweat gland, which secretes a watery fluid called sweat. Primary function of sweat is thermoregulation through cooling effect, because body temperature is maintained, which gives a cool effect when we do fast activities. Sweat is released through these glands, and which gives the cooling effect. That means the temperature of the body is maintained. So it also helps in removal of some of the waste like NaCl, small amounts of urea and lactic acid also. So through sweat glands, these along with water, it excretes these wastes like NaCl and urea and lactic acid in small amount. Second type is sebaceous glands or oil glands. These are called as, they eliminate certain substances like sterols, hydrocarbons, waxes through sebum. And what is the function of sebum? Sebum provides a protective oily covering on the skin, which is very important. Small amount of nitrogenous waste are also eliminated through saliva. Remember this, you might not be knowing. Through saliva also, these nitrogenous waste are eliminated in a very small amount. Now whatever the disorders are remaining, that we if you want, I will continue. Otherwise, we will be having little revision also about the part which we have taken.